from Texas A&M University in 2011, and then continued with her postdoctoral training at the University of Texas MD Anderson Cancer Center. And Elisabeth's research group studies mechanisms for fatigue and depression in relation to cancer and inflammation. And today we will learn more about this while listening to her talk, Mechanisms Underlying the Persistence of Cancer-Related Fatigue. So thank you so much for being with us here today and for sharing your work with us. And the stage is all yours, so you can share your screen.
Thank you. I'm excited to share this research. I'm focusing on, make sure I'm in presentation mode. All right, let me know if you guys can't see. I'm excited to share my research with you. We're focused today just on cancer-related fatigue, as my topics tend to be a little bit variable, but this is a topic near and dear to my heart that I've been working on for probably a little over a decade now, and so I'm excited to talk with you about it. See if I can progress. There we go. So in general, I'm going to kind of give the scope of the problem. The scope of the problem is probably what drove me into this area of research, was when I started to really understand the problem better. I'll talk about how I study it in murine models, mice, and then kind of the working model I'm developing kind of as I try to conceptualize the problem and how we might approach it a little bit better. So if you kind of think, and these statistics are limited to the United States, as that's what I was most easily able to find, but what I want you to see is the growth curve that we're seeing. So when this came out, this was in 2019, but now we're at about 18 million cancer survivors in the United States alone. And in the next 17 years or so, we expect a growth trajectory of about 25% more cancer survivors, which is a wonderful problem to have, I suppose, to phrase it, where people are surviving cancer better, but we're also seeing an uptick maybe in some rates of a few types of cancers that may also be contributing to this. And if we try to look at it at a worldwide scale, that probably means currently we're at about 32 million worldwide, and I would actually expect that the percent increase would be higher, at least 25% also on a worldwide scale. So we're looking at tens of millions of people throughout the world who have survived cancer. If you look at the age distribution of these, most of them, about 2/3, are 65 years of age or older, because cancer is a disease associated with aging. Our risks go up as we age, but that also leaves about 1/3 of people who are below the age of 65, meaning there's still a large lifespan expected post diagnosis. And if we kind of try to break that up to the number of cancer survivor years after diagnosis, we have a good spread of people living 30 plus years after their diagnosis, meaning we're living with these symptoms and the side effects and the consequences of our treatments for a very long time, potentially. And so this allows us now the luxury to start considering quality of life considerations for patients and how we can improve the quality of life post entering into a survival phase. In one study by Shuling Shi, a colleague of mine at MD Anderson when I was there, she looked at almost 5,000 cancer survivors. And if you looked among those patients, 92% reported symptoms related to their cancer or treatment. 29% of these were classified in having high symptom burden post treatment. When she kind of started to parse what created risk for these high symptom burden, metastatic cancers, continued treatment, so a lot of cancer therapies need to continue well into their survivor phase where you are receiving treatment for long afterwards. Comorbidities, different diseases, younger patients who actually seem to be at higher risk for a higher symptom burden afterwards. And those from medically underserved populations seem to be at highest risk for having this more impaired quality of life. And if you looked at what was affecting quality of life the most, depression, pain, and fatigue were among the top 3 factors impacting them. And fatigue consistently across studies and across a number of areas comes up as a very high factor. And what catches my attention about fatigue is that we have the least understanding of its mechanisms and currently the least number of medications available for treatment of fatigue. So there are some things on the market, and I will say we're not there with depression yet, but we have options available with pain. There are options, not all optimal options, but options. With fatigue, we're a little more limited with our toolbox of what we have. When looking kind of at these patients and across different cancers and things like that, patients under treatment, you can see compared to patients that are surviving, and you can see that in the survivorship phase, you do see a lessening or a reduction in the number of people with severe and moderate symptom burden, but that still remains relatively high. And if you look and divide those survivors up between those that are less than 5 years or more than 5 years post-survivorship, you can see that it really does persist for a significant amount of time. And if you look at this study and across a variety of other studies, kind of trying to quantify this, we would say that around a third of patients experience a persistent fatigue phenotype after their therapy. And if you're thinking we're in the, 35 million people, now we're talking about 10 million people that are in need of some sort of intervention. It's probably, maybe a little late, but probably important to kind of define what we mean when we talk about cancer-related fatigue. And this is probably the most commonly accepted definition from the National Comprehensive Cancer Network. It's the distressing, persistent, subjective sense of physical, emotional, or cognitive tiredness or exhaustion related to the cancer or cancer treatment that is not proportional to reasonable activity and interferes with your usual functioning. And that's a long-winded definition. But what we should notice there is that it's a complex. It's not just one thing. It's physical, it's emotional, it's subjective, and it interferes with things. And that sleep doesn't merely fix the problem by itself. So not proportional to activity. So going to bed, getting up, waking up fatigue for some of these people is their experience. I think it's valuable also to kind of look at what patients describe. This is from one study. I spent time in clinics also listening to patients, and they describe things like listlessness, sluggishness, apathy, indifference, heaviness of their body, when they think about kind of what is impacting them. And if you kind of try to categorize these like Moeller and Walgren did, you can kind of see that there's something about the experience of fatigue that patients like they describe. They describe the stress associated with it, the emotional burden associated with it, the lack of initiative, but they also describe the consequences, that they begin to socially limit themselves. They affect their quality of life. They affect their self-esteem. And so it's a multifaceted problem. And there are currently no FDA-approved treatments for it. So within the United States, we have nothing that's on the market to treat this. What is currently prescribed for most patients who are talking to their physicians about fatigue, and I will say a lot of patients fail to even mention their fatigue phenotype to their physicians for a couple of complex reasons. Some believe that it's normal and that's kind of just what you have to expect. Some of them are afraid that of what it means to be fatigued afterwards. And so maybe they are worried that that infers something's going wrong with the disease process, or that their treatments will need to be changed, they'll be put on different medications. Some just don't feel that their doctors are the appropriate people to talk to about this. And so a lot of people don't report. But when they do report, generally what is recommended is, well, energy saving techniques. So use your the most important things of your day, front load into the day when you have the most energy and you're feeling your best. Exercise, which you should watch patients' eye rolls when you look at them and they've just told you, I can barely get out of bed in the morning, and you're thinking, have you thought about getting up and exercising? But really the data is really quite convincing that if you can get patients moving, that it actually does improve does improve function and does reduce fatigue levels. So if you can get patients exercising, it does help a lot. Sleep hygiene, obviously, nutritional consultations, mindfulness practices have been shown to be beneficial. Cognitive behavioral therapy is sometimes used. And all of these can be very, very beneficial. It can be a multi-pronged approach, certainly not one strategy can be used, but these are also complicated and relatively expensive approaches for the most part. Cognitive behavioral therapy isn't necessarily available to all patients, and we already discussed those that are medically underserved are more likely to experience fatigue and may actually have less likeliness to have the resources to have things like CBT. Psychostimulants are sometimes prescribed. So methylphenidate is probably one of the most commonly prescribed psychostimulants for cancer-related fatigue. It's obviously an off-target prescription. Most of the systematic reviews and most of the analysis I went through show very mixed findings. So the large meta-analysis here doesn't show any definitive effects. I'm not to say that it couldn't be beneficial, and I think there's likely a subset of patients who may benefit from psychostimulants, but it is certainly not going to be the kind of solution, the way to solve the problem as a whole. And I think that's because we just don't really understand all of the mechanisms well enough to have a really appropriate therapy yet. What we do understand is that our mechanisms are multifactorial. When I started into this investigation, I was very, very much focused on the central factors, particularly inflammation. The hope that when we entered into this field was that if we could measure that this was inflammation, we could treat inflammation, we know how to treat inflammation reasonably well, and that we could treat the process. There are a really beautiful array of studies showing a relationship between both peripheral inflammation and the development of fatigue trajectories. There's some data on some levels of central inflammation being associated with the development of fatigue, and it makes a lot of sense when we think about sickness behavior and inflammation-induced sickness behavior. That's very much similar to the fatigue phenotype we described, and so it made a lot of sense. For us, what we're seeing is that while inflammation may have a role, it doesn't seem to be sufficient, at least in any of our Murine models, to fully treat the problem. And so inflammation may be a component, but not the entirety of the story. It may be that the drugs or the tumor itself cause damage to the central nervous system, so maybe neuronal damage is involved, and we've begun to kind of consider altered cognitive processes, and I'll get more into that throughout the talk as a potential central mechanism. Peripheral mechanisms are something I've also begun to appreciate a little bit more. Many of the patients who experience cancer end up with some levels of cachexia, which is a muscle wasting. And it isn't a hard stretch to see how weakness of the muscle wasting would cause muscle weakness, and muscle weakness could lead to fatigue. And so these are intrinsically linked phenomenon that are hard to disentangle. And so when some people are describing their fatigue, it could in fact be part of this muscle wasting phenotype and not necessarily directly a central process. Also, energy balance issues have also come up as really probably critical to understand. If you think about what's happening when you have a tumor growing within your body, you have this essentially metabolic sink, this energy utilizer. And we generally don't increase our eating to compensate the increased energetic demands of the body. And in fact, due to nausea, vomiting, and changes in appetite, there's actually a loss of eating usually. And so you have more energy expenditure and a loss of nutritional intake, and so you have this energy balance, or this negative energy balance. And so what happens is either you reduce your activity to spend less calories, or you end up losing muscle mass and wasting away. And so fatigue could clearly fit into the let's reduce our movement kind of ideas. And different mechanisms likely underlie the initiation and maintenance of fatigue, so that a treatment that's good at one point may not actually be the optimal treatment at another point in the process. And we're starting to kind of begin to look at that with a little more finesse. And I think that this will require a combination of clinical and preclinical approaches. So while I would say that there's a lot we can learn from approaches that I utilize, there's some limitations to using mouse models as well. Some of the limitations is that we're limited to observable activity in our mice. And some people have questioned whether we can genuinely study fatigue in a murine model. I would say that there's a lot that we can do. It's imperfect. We study things like activity. We can study how much they move and how fast they move. We can use things like running wheel or burrowing activity or home cage activity. We can study their motivation, so how much they're willing to work for rewards, kind of this lack of initiative, apathy kind of phenotype. And then cognitive fatigue, we can probably get to some elements of that through attentional tasks. Obviously, there's a lot of interplay between these type of tasks, and they're likely to, if you have a motivational deficit, impact all levels of observable behavior. We're also limited to murine models, limited to tumor that are from a mouse rather than from a human if we want immunocompetent mice. So if you want to study a tumor in a mouse, you need to have a tumor that can grow there. And I need my mice to have an immune system on board if I think that the immune factors are playing a role. And so I can't use human tumors. So that's another limitation of what we work with. So we work with mouse tumors rather than human tumors. But what some of the strengths that this allows for us to do is to parse tumor and treatment effects, things that we can't ethically do as well in humans, and to evaluate brain changes in a little more direct of a fashion. And so I think that's what our models can add to the picture. In general, what we do is we grow cells in a dish. We use a syngenic, meaning a mouse tumor that can grow, and then we heterotopically implant it into the mouse. When we heterotopically implant it, we mean we're putting it where it doesn't necessarily belong, but where we can monitor it well. We want to monitor the growth. So we put it into the flank so that we can see it where it's at. Because if we put it into, for example, if I use a lung cancer model and I put it into the lungs, monitoring the growth in the lungs will be very challenging and makes it a little more complicated and a messier model. I frequently use a mirror tumor model, which is a model of head and neck cancer, and the LLC tumor, which is a lung cancer model. We then can later go on and treat these mice with chemotherapy or radiation, or not treat them. We can also treat with chemotherapy and radiation apart from the tumor. And then we can monitor their behavior and collect tissue. So when we look at cisplatin, which is the chemotherapy agent I use most frequently when studying these, just because it's ubiquitously used across a wide variety of cancer types, what we can see is that both male and female mice, when treated with cisplatin, show a strong fatigue phenotype as assessed by running wheels. Running wheels is one of my favorite ways to do it because it's very passive. It doesn't require as many manipulations of the mice, and we can see their behavior in a more natural setting. They also like to run a lot. So it's works on some of their natural skill sets. So what you can see is where those arrows are, where I treat them, and you can kind of see the scalloping to the point where they kind of holds them down at a low level for a long time. What Sindhuja did for her honors thesis recently was start to look at this with a little more finesse. And so one of the questions we've had is kind of, well, what does this look like? If we try to really look at this reduction in running, are they running less wheel rotations? Are they running less often? Are they running more slowly? Kind of what is this? And so what she showed is that in response to cisplatin, we observed a reduction in the amount of time they ran on the wheel, as well as the speed that they ran at. When we think about changes in running speed, this points me towards a central process. It kind of points me towards the literature on vigor, which suggests that this central dopamine kind of governs a lot of our speed of movement. And so it makes you to think that cisplatin may be inducing some of this fatigue through a centrally mediated mechanism. And one of that, a lovely paper that I was a minor collaborator on by Xu et al., looked at some of the things that happen in response to cisplatin. We see a reduction in neural progenitor cells in the brain. We see a loss of arborization as measured by a coherency index. You see changes in mitochondrial structure such that they're more atypical, and mitochondria kind of driving the energy of most of our neurons are critical. And not only are their shapes altered, but their function is altered. So if you put them in a test where you ask them to perform at their maximum capacity, these mitochondria are not able to don't consume as much oxygen. So that's what the OCAR stands for, the oxygen consumption rate. So they don't consume as much oxygen, meaning they're not as effectively generating energy for the system. And so cisplatin seems to impact the brain on multiple levels. And in this study, there was an agent called PFT-mu, which was able to reverse some of these changes by protecting mitochondria. It is acts at the p53 area of mitochondria to protect it from cisplatin-induced damage. It's a messy drug. I wouldn't use it in a clinical setting, but in terms of experimentally, it demonstrates that these cisplatin-induced changes are largely derived from mitochondrial deficits. So we've got these mitochondrial central deficits that may be largely contributing to cisplatin-induced changes. And then we wanted to start, when we look at tumor-induced changes as well. In response to tumors, we see a progressive decline in activity in our mice. So you inject the tumor, these mice are not treated in this context, and you see a progressive decline in their activity. If you look at those lines, that 9, 16, and 27-day point, you can kind of start seeing what we measure in terms of peripheral inflammation and central inflammation. So while we start to see declines really pretty early on, it isn't until later on that we begin to see these development of peripheral and central inflammatory cytokines, suggesting that something else seems to be driving that early fatigue-like behavior in our animals. And if we look at these, what we can see is that this was also Sindhuja's honors thesis. She showed that rotations are reduced, run time is reduced, but the run speed is not, suggesting maybe this is more of a peripherally driven phenomenon rather than central like we were seeing with the cisplatin. And if we kind of trying to take a very, very complex study and pare it down into something kind of digestible, is that what we observe if we analyze kind of the metabolic kind of system of our organism after we implant a tumor is that we see this increase. We see this just, well, the Cori cycle is a really important component of how we normally function, which means that our muscles produce a lactate. Lactate is taken up by our liver and is converted into glucose. The glucose goes back into the bloodstream to be reutilized. And that helps us to have a lot of metabolic flexibility. When you implant a tumor into the animals, what we see is that our livers are less capable of engaging in gluconeogenesis and engage in a high level of glycolysis, which is a different way to process energy. It's also a method of processing energy that's associated with more reactive oxygen species and maybe oxidative stress type phenomenon. We see a buildup of lactate in the muscles such that that lactate doesn't seem to end up going into the bloodstream to be taken up by the liver. We also see a reduction in glucose in the bloodstream, likely because the tumor is taking up whatever glucose is available to feed itself. And so what we see is this overall dysregulated metabolic phenotype throughout the periphery, creating this kind of metabolic sink that I was talking about before, that the tumor just creates these problems that drive this reduced running. And this phenomenon is heightened if you ask mice to run, or if you take their food supplies away, this process is even more exaggerated. We can say also that if we look at this, we tried reversing this with a variety of anti-inflammatory agents because there was some suggestion in the literature that and IL-6 from the tumor may be what's driving these altered processes in the liver, and that may be driving these larger term changes. We tried reversing with an IL-6 neutralizing antibody. We tried with IL-1. We've tried with a variety of other anti-inflammatories, and we're unable to reverse this metabolic phenotype. Not to say that we don't get a lot of IL-6. The liver does produce a lot of IL-6 in the tumor model. It just doesn't seem to be that blocking it doesn't seem sufficient to reverse the phenomenon we're observing here. One of the questions I had, and one of the questions that continues to spark my interest, is the idea of what is it, if it's not IL-6 from the tumor, what is it from the tumor that is communicating to the rest of the body to kind of create this whole altered metabolic system that is adaptive for the tumor, but detrimental to the host. I think of the entire host as the tumor macro environment, and the body is trying to find a way to do that. And there's probably a variety of things. Tumors release a variety of things. One of the things that caught my attention recently was something called amiloride. Amiloride is a drug that's able to reduce exosome release from tumors. So tumors release exosomes, which are these little tiny packets of information that we thought were mostly that cells were just eventually kind of throwing away, kind of like the trash of the cell. We don't need it anymore, get rid of it, don't get rid of it. But what it appears to do is that these little packets of information get released by cells to communicate. And they may be that exosomes are an alternative communication mechanism between two cells. Tumors release a bunch of exosomes, and this drug can seem to prevent much of that release, and it stops cancer-induced cachexia. So I had one of my students start looking at this effect in our fatigue-like behavior, and we see a subtle effect. It isn't as clean as we'd like, and we're still kind of exploring it, but what we can see is if you look at the tumor, which is the solid red line, and compare that to the tumor amiloride, which is the red dotted line, you can see that the amiloride attenuated our running deficits, such that with the amiloride treatment, we ended up with a better overall activity profile, so a reduction in fatigue. So it may be that things like exosomes are involved in this process. The amiloride also reduced liver IL-6 expression, and we still obviously have a lot of work to do ahead of us to try to see how much of the metabolic phenotype can be prevented by stopping these exosomes, but exosomes may be one of the communication mechanisms by which tumors communicate to the rest of the body. I would certainly not argue that it's the primary or the only, but maybe a critical one. In thinking of, and I guess adding more complexity to the story, it also seems to be tumor specific. So I would have loved, from my non-tumor biologist in me, likes to think a tumor is a tumor is a tumor. You put one in, it should do all the same thing. What I'm learning is that that oversimplified approach is probably not going to work. One of the first positive results I saw with attempting to reduce tumor-induced fatigue was with my Lewis lung carcinoma. This is in the red. I should have labeled that, my apologies. Is when we used TLR4 knockout mice, we ended up with an attenuation or a reversal of their fatigue-like behavior, which was really exciting for me because this started to point me to all kinds of things that ended up not being quite accurate. But when I take that same manipulation into another tumor model, the mirror tumor model, we get a very different phenotype where our TLR4 mice perform just like wild-type mice, suggesting that different mechanisms are involved at different tumor types. What I actually think is going on here, and I didn't bring all the data to try to justify my explanation, so if you want to discuss it with me, let me know, is the LLC tumor, the red tumor in this case, is a cachexia driving tumor. So there's a large cachexia phenotype associated with the LLC tumor. There is not a large cachexia phenotype associated with the mirror tumor, the one in blue. The TLR4 knockouts appear to be helping to protect against cachexia and the muscle fatigue that comes with cachexia. That is not what's driving the fatigue-like behavior in our mirror tumor model, suggesting that that as we test these, it'll be really important to test the generalizability of all of the different interventions we identify, because one may work beautifully in one context, but not generalize necessarily to the next context. And so what I would suggest is that in our LLC tumor, which is also the tumor in which the exosome study worked, that there is a large cachexia phenotype that may be driving our fatigue behavior, but that there's a different fatigue driver in the mirror tumor model. So just to keep things complicated. All of that to set us up for fatigue and survivorship, which was kind of the aim of this. And I apologize for putting so much of a preamble there, but what I wanted to start understanding was what causes it to perpetuate for so very long. And what we were able to do with our mice is find in this model, this is with the mere tumor model, in which we treat them with 4 doses of chemoradiation. And what we're able to do is cure the tumor, essentially. So we create a mouse that had a tumor, had a course of treatment, and now is tumor-free. And our tumor-free mice end up having a fatigue phenotype that's measurable for at least a month out. And so we thought we had a really nice model to start working with to talk about fatigue and survivorship in our mice. One of the things that's really interesting about this is that there were a few things that made it more or less likely for us to see this strong fatigue phenotype. And one of it was what we did during treatment. So I collaborated with a lab out of Sanford, and what they used when they irradiated their mice, they put their mice under anesthesia. They used a ketamine-xylazine combination. When I didn't, I just physically restrained my mice. My mice showed more fatigue than theirs. So we ran a systematic study, and we showed that if we treated with either isoflurane or ketamine-xylazine at the time of the treatment. So when I treat my mice, I pick them up, I inject them with cisplatin, and I throw them in the irradiator. I place them gently in an irradiator. And if we treat them at that point in time with an anesthetic, we have a better recovery trajectory during survivorship. Well, I started thinking about what these drugs can do, and there's a literature suggesting that they can be involved in disrupting memory consolidation. What if there's a component that is learned during the treatment that is critical for the persistence of this fatigue and survivorship, is what we started to speculate. And that something about the experience of fatigue during cancer therapy is what helped it to perseverate. And so what we did is a study that in which we either allowed our mice to stay with their running wheels during treatment, or we removed their running wheels during treatment. So we trained them on running. It takes about 2 weeks for them to get up to maximum running speed, and they remain very consistent after that. We then either left them with their wheels or we removed their wheels. And then we returned their wheels a few days after their last session of chemotherapy and chemoradiation, and we put those back in. And what we saw was that while it's subtle, it's still there, that there's a protective effect of not having their wheels, which is very counterintuitive. For exercise is generally accepted as a really beneficial, positive aspect for cancer patients. I told you that when people are fatigued, it's the best evidence-based therapy for fatigue. And now I'm trying to suggest to you that running during treatment is negative. And I don't know if that's exactly what I'm saying here, or not what I'm exactly trying to say. But what I'm saying is something about experiencing fatigue during treatment, which is something we cannot fully eliminate from patients, but can do so more from a mouse perspective when they're in a very deficient environment, essentially. If you don't put a wheel and you've individually housed an animal, it doesn't have much to do. And so its fatigue experience is relatively limited. If you look at it, this is day 49, where you can kind of see the trajectory. So the dotted line are the mice that didn't have wheels during treatment, and the solid line, the solid red, is the ones that had their wheels during treatment. And you can kind of see that they just don't get as many rotations in per hour when they had those wheels through the whole time. And it's quite a prolonged phenomenon. We wanted to see if that was related to more toxicity in this process. And so what we did is we looked at, cisplatin is known to create kidney toxicity in particular. So we looked at the kidneys of these animals. And what we show is that actually those without wheels had probably the most severe toxicity phenotype, and it was attenuated or blunted with running. So running blunted the toxicity profile in terms of the kidneys. Urea is also we measure that in plasma. It's upregulated when the kidneys aren't functioning as well. And so running, in fact, improved the toxicity profile without improving our behavioral profile. So what we'd say is that exercise is certainly not bad on the toxicity front. It's actually beneficial. So we began to think about what fatigue might be, and if it's conditionable, if it's something that's learned in process during these sessions. And so we thought, okay, we know chemoradiation produces a fatigue phenotype. So we started to pair chemoradiation with a very distinct olfactory cue. Bergamot oil is what we used, randomly selected, no actual good rationale for why we chose that one. And we asked then if we later expose them to the bergamot oil, would we end up with a experience of fatigue? And in fact, we did. So what we did is we did this for 3 sessions of CRT. Then we brought the oil back into their cage after they had shown over about an 80% recovery of their fatigue. And we get a 1-day drop again in their running, suggesting that they built an association between cues in their environment and their activity levels, which would suggest to us that there is a cognitive circuits involved in the maintenance of fatigue, and that if we could understand how these cognitive circuits work and which ones maybe are maintaining this process, we may be able to develop interventions that focus on those. So our main conclusion based on that 2023 paper was that the associative learning processes modulate the persistence of cancer-related fatigue behavior. And this has started me to start thinking about what associative learning is, what it means in terms of the brain, what circuits guide it. And I started thinking a lot about habit behavior and what creates habits within our brain. So when we think about habitual behavior, these are kind of ways we automate ourselves to be efficient, as opposed to goal-directed behavior, which is very focused on rewards. And different brain regions in the striatum have been associated with promoting these. So the dorsolateral striatum is most associated with habitual behavior, whereas the dorsal medial striatum, goal-directed behavior. And a variety of regions, such as the orbital frontal cortex, seem really important in helping us to switch between the two, because it's really important we're cognitively able to switch back and forth. And so in reading, I started coming up across a lot of interesting literature that would suggest that, and this is probably literature I was familiar with once and it just kind of fell from my memory, was that stress promotes habitual behavior. If you stress people out, most of these were done in clinical populations or healthy control populations, a few in mice, but if you stress people out, we're more likely to rely on our habits than we are to rely on goal-directed selection of choices. And if you think about fatigue from that perspective, I started to wonder if a very similar relationship may exist. So in general, what they show is that habits, which are what allow for us to have a really complex set of behaviors that don't require as much cognitive effort, which is exactly what we should need in fatigue. When we are very fatigued, we're trying to save our cognitive resources. So goal-directed, thinking of it as more energetically expensive for the brain, has a response outcome contingency. We move into these habitual control, which is a stimulus response, which requires less cognitive resources. We need to be able to switch back and forth frequently. Stress makes it harder to switch back and forth. And we started to wonder if fatigue would do the same thing. If maybe what was happening during fatigue is that we were shifting ourselves into this habitual circuit of the brain, and that we were having trouble pulling out of that circuit because of the fatigue itself, making us be more dependent on the habitual control. So we decided to try to assess whether the drugs that we use to induce fatigue also can induce changes in habitual behavior. And so the way these studies kind of tend to lead, there's a couple ways to do this. We decided to do a devaluation procedure. In a devaluation procedure, you train an animal to lever press for a reward, or nose poke. In our case, we use mice nose poke better than they lever press. So we nose poke for a reward, and then we devalue that reward by giving them plenty of the reward prior to the task. So they They don't want the reward anymore. The reward is less valuable to them because the way our brains process whether something is rewarding has a lot to do with our particular level of satiation. And then we put them back into the test and say, okay, now that you've fully satiated, you've had your reward, how do you perform? And animals that are habitual performers tend to continue, in this case, lever pressing, and in our case, nose poking, in the face of not actually wanting the reward, because there's these response outcomes. They see the stimulus, the nose poke or the lever, and they act on it kind of without thought. I don't know, maybe I live in this kind of mild level of continual stress, but I'm the type who will walk by any candy bowl and throw a piece of candy in my mouth without ever thinking about it, even without wanting or enjoying that, out of habit. We have habits drive so much of what we do. And so we asked if cisplatin could promote this habitual responding, and that's exactly what we saw. That we saw a change in our nose poke number was much less. So our vehicle mice showed devaluation, that when you put them in a satiated situation, they showed much less responding. Whereas our cisplatin were actually very resistant. They kept working harder in the task, even though they didn't need to, because they seemed to be in this habit circuit, which is a very surprising and counterintuitive. I very rarely am able to make my fatigued mice work harder than my control mice. And so we were really excited to see that data. We've just finished a replication and we're pending analysis. So I don't have the data yet, but hopefully by next week, I should have it analyzed. And so kind of what we are at right now is kind of our working model. We're thinking about the concept that the cancer treatment through various mechanisms of altering the central nervous system, of altering brain mitochondrial function, and our cancer itself by introducing these really pronounced metabolic changes within the body, produce this fatigue phenotype. Sorry, I'm not moving forward well. The fatigue phenotype starts causing us to become more dependent on habitual behavior. And this to allow for those cognitive resources to be devoted to things that are more important at the time. And so the fatigue starts promoting that habit behavior, and that the habit behavior then continues to promote the fatigue's maintenance. And that in order to help, theoretically, if this model holds any weight in maintenance of fatigue, we need to break this habit circuit. This may be why treatments such as exercise or cognitive behavioral therapy are able to be effective, is they help pull us from the part of our brain that drives our habits towards more goal-directed behavior again. I will say that there's also large reasons to believe that residual side effects of cancer-related toxicity are also involved on a variety of fronts. So I'm not to suggest that this is the only way, but that this is involved in the maintenance, and that for some individuals, being able to better understand this loop and being able to better understand kind of how fatigue could be maintained might allow us to have a better sense of, to design better therapies that may work for a subset of patients during the survivorship phase, and that different interventions may work, and multifactorial interventions are likely necessary that both treat any underlying toxicities that are remaining, as well as any cognitive processes that are promoting the persistence of this behavior. So that is what we're currently starting to explore, is the neural circuits that are involved in this, and kind of where that, where we might be able to intervene and how we might be able to intervene. So that's kind of where I'll leave you guys today. I wanted to thank my lab group, particularly my two graduate students, Haley Rogers and Kayla Gilly, who will be defending her dissertation later this afternoon. Some of my other collaborators who work on the cachexia work with me, Dr. Michelle Law and Dr. Michael Wiggs, and certainly my neuroimmunology laboratory at MD Anderson, and particularly Robert Dancer, who's been really critical in kind of forming, helping build this body of research and helping me move forward in my own independent career with it. Thank you so much for listening. I'm happy to take questions, or anything you have, comments, questions, anything. Thank you so much.
Great. So thank you so much for this very intriguing talk, Elisabeth. The audience, you can ask any question by pressing the Ask a Question button and right there. But I can start and just ask. So I was intrigued by this, that you can very quickly in the tumor model see that it affects the wheel running. So I was wondering if it's the same with these more like reward motivational tasks, that you also see it so quickly, or is it something that comes later with this type of tasks? Is it more linked to inflammation, for example, or?
That's a really good question. The motivational tasks. So if we look at just like nose poking with our animals, we actually don't see much of a difference. And it's one of the things that I think continues to perplex me, is why we, let me end my share for a second. Yeah. One of the things that I continue to stay perplexed about is how little direct motivational shifts we see in our animal systems. And I think that's partly because our mice are a limited system in terms they don't understand their diagnosis. They don't have the psychological stress of a diagnosis. I think that part of why we see such a strong motivational component, at least I hypothesize, it has to do with the psychological stresses associated with their cancer diagnosis that we do not replicate very well in our animal models. So I think that it's very likely in humans that those motivational components would be a slower developing and might very well have an inflammatory component. We know that if we inject our tumor-bearing mice with a little bit of sub-threshold LPS, we can get them into a depressive phenotype, but we can't get them very depressed. But normally they don't sit depressed. Well, many of our patients do report depression. So it's one of those limitations. Does that sort of answer your question?
My mic was off. Yes, thank you. So we have a question from Sita. Do you think that the effects of cisplatin on causing fatigue come from its systemic effect on multiple organs or directly on the brain, or that it comes from its actions at the tumor level?
I think that's a multiple part question. So yeah, so I think that when you're giving it in a non-tumor bearing animal, so taking the tumor out of the picture, I think there's very many reasons to believe that it's going to impact mitochondria on multiple fronts. So we know the brain mitochondria seem to be affected, and that if you can protect them, you can create some level of protection from the fatigue, or you might be able to protect that fatigue phenotype that way. But I would say that it's likely you also have peripheral effects. So cisplatin is a DNA chelating agent, and it seems to bind to the mitochondrial DNA at quite a high rate, which can cause mitochondrial damage on multiple systems. We also see mild levels of cachexia in response to cisplatin treatment as well. So while my wheel running data would suggest that there's probably a central phenotype involved, that doesn't in any way take away the idea that it could be a peripheral phenotype as well. So I think that it's probably both, but I do think central contributes. When there's a tumor, we seem to see that you end up with two processes. By suppressing the tumor growth, you protect against the tumor's induced effects, and so cisplatin actually improves outcomes for our tumor-bearing mice rather than reduces outcomes. So if you let the tumors just go unhindered, that's a worse outcome than tumors with cisplatin. But the idea that the cisplatin acts to cause cell death within the tumor, potentially creating more inflammation, is certainly a very viable hypothesis, not one that I've been able to tease apart yet, but I can see that that's a very interesting idea that would take some work, I think, to tease out.
Yes, great. And then we continue with a question from Rita. Hi, thank you for the interesting talk. Maybe I missed it, but does exercise restore the mitochondrial deficits in cancer patients?
That's a really great question, not one that I have explored personally. I do believe that there is a literature showing exercise can improve mitochondrial function. I just don't believe it's been integrated into the cancer fatigue literature yet, but I do think that's a very viable idea for one of the mechanisms by which exercise may be improving. I think there may be a clinical study in which they did maybe PBMCs and trying to look at their mitochondrial function in response to exercise, potentially showing effect, but that may be, but I can't remember that reference at the top of my head, so it may be inaccurately remembered. But yes, I think that's a very viable idea and a very reasonable hypothesis, one that I can't verify yet, though.
Is exercise used like a lot in the cancer treatment today? Like, is it specifically used for some types of cancer and so on in the like during the treatment and so on?
During treatment, we give very mixed messages to our patients. So during treatment, most physicians will promote energy conservation strategies rather than exercise, but telling patients to remain active when they are capable and within bounds of reason. It depends on the stage and the severity of their treatment and the severity of their cancer. So there's a lot of factors that put into play. There is evidence that suggests that exercise can be really good for promoting the vascularization of the tumor, which promotes getting chemotherapy into the tumor. And so that is a really interesting idea to suggest that exercise could help with the efficacy of your therapy by promoting angiogenesis there. But I think we're hesitant to directly prescribe too much exercise to patients, knowing that you've essentially, where some of these people are just pumped full of toxins and their bodies are so weak. And so exercise when they can, I think, is what's generally recommended. Once we get to a point where patients are more towards the recovery trajectory, that's when we really start pushing, at least from my experience, and I am not a physician, and I have never actually made any advice to any particular cancer patients. Let me just clarify there. But I shadowed in a fatigue clinic for a while. And in doing so, I would watch that once they were into a survivor phase and they were capable of taking on more, that's when she would start pushing them to exercise pretty across the board. The patients she saw the most were breast cancer and head and neck, but I have no reason to believe that it would be specific to those cancer types.
Okay, so if we have no more questions from the audience, we will thank you again for this great talk, Elisabeth, and thanks to everyone in the audience. So next week we will have our last seminar for this semester, and it will be with Dr. Michael Burton, and he will talk about neuroimmune crosstalks in pain and inflammation. So we will, maybe there it happened something. Maybe we have time for, okay, we will continue with at least one more question here. I missed them, sorry. Okay, so we have one more here. Regarding the metabolic changes associated with Cori cycle you mentioned, I guess that will lead to changes in circulating metabolites. Yes. Can this lead to fatigue in two ways, one directly in muscle function and another in neuronal function? So this was another question from Sita that we will have time for as well.
That's a good question. I think that it could create fatigue through multiple mechanisms. So yeah, I think the alterations at the muscular level are really important. I think that the circulating levels and alterations of glucose levels and other metabolites that communicate our energetic status to our brain is probably really important. And I don't know exactly what that, and I know that's not exactly the question she's asking, but that there's signals between our body and our brain that tell us how much energy to expend. And those signals are probably found within circulating metabolites. And what was the system that she was suggesting? I've missed that one. I think we have you on mute. Lena, we have mute. You're muted.
Oh, sorry.
That's my fault. I was trying to remember.
Yeah, so I guess this will lead to changes in circulating metabolites. Can this
Yes.
lead to fatigue in two ways? One directly in muscle functions and another in neuronal functions.
Neuronal functions. I would suggest that's probably true. I don't know as much about that, but I think that's very possible.
Great. Okay. So one last question then. This one is from Sylvia. Great talk. Do you know if a higher mental burden also acts in a similar way to increase exercise in changing fatigue?
That's an interesting way. So for like a cognitive fatigue, if pushing cognitive processes could, we know cognitive training can help in other contexts, more protection than treatment, but it's very possible, especially if those exercises somehow promote utilization and shifting between those two system levels. I think that could be very possible. And I think that the optimal type of treatment strategy would employ something like that, and that's where I think cognitive behavioral therapy could come into play, with trying to help find ways to kind of alter the cognitive processing at that level in the brain to help move people over.
Great. So sorry for being a bit quick there and missing two questions, but now I think we have had answers to all the questions, so that's great. Thank you so much. And so again, this we will have the last seminar next week. And we will now move to Zoom for a Zoom question and answer. So if you're interested in like discussing a bit more, asking some more questions, you can join us. I will send the link in the chat now. So we have that there.
Wonderful.
So we will continue there. And thank you so much to the audience and to Elisabeth again.
Thank you.